Good. So, no, once more, welcome to everybody. I have set up this Zoom meeting in such a way that only the speakers are able to unmute themselves and speak. This is not because we don't like other people to say anything. This is more because it often happens that somebody forgets that they're unmuted and then all of a sudden start speaking through a presentation, and that can get quite embarrassing and sometimes just annoying for people listening. So that's the only reason that everybody's set up to come in muted and not be able to unmute at this point in time. If it becomes necessary at some point that we want a broader discussion and people want to come in, I can easily unset that feature within 2 seconds, so that is not a problem. In the meantime, therefore, you do all have access to the chat, and feel free to use the chat to chat, but also to write questions to the speakers, which you can even write during their presentation, and I will keep a little eye on them. And then at the end of the presentation, I will also go through, look for questions there, and talk about them. So the presentations will be roughly 25 minutes each, with a good 10 minutes for discussion and questions after each presentation. So before we start, I'd like to welcome you to this Spring Symposium, which is, I don't know, the second or the third now that JB Johnson Club has run. Third, sponsored by Karger and Brain Behavior and Evolution. And I would like to hand over briefly to Spock, who's going to speak on behalf of the JB Johnson Club before we go back to the actual symposium.
Thank you very much, Tom. I really appreciate this opportunity. Again, this is our third Spring Symposium, so we welcome everyone here. If this is your first time here, please think of the JB Johnson as a home for your comparative nervous system research. Just a little bit of housekeeping. I want to remind folks that if you're not familiar with brain behavior and evolution, this is perhaps the main forum, and I should say our official journal for the JBJC. Please think of this as a forum for you to publish your work and that of your students too. Tom, you can go on to the next slide. Again, as part of the JB Johnson, I want to remind folks of our upcoming in-person meeting. This is our 35th Cargo workshop and our 43rd annual meeting that will be in Washington, D.C. during November 9th and 10th, just prior to the Society for Neuroscience meeting. We will be at Howard University. This year's workshop is organized by Dr. Carmen Falcone, and it's on the evolution of glia across invertebrates and vertebrates. Again, that call for abstracts will be going out within the next day or two. And then again, the abstract deadline is usually a couple of months beyond that, 6th of July. So please look out for those emails. And if you aren't on our list and you've just joined recently, please feel free to reach out to myself or any of our members. Tom is another one. We can put you in touch with me and we can help you along that path. Next slide, Tom.
That's me already.
That's it. Anything else? I think there's one small one I just want to remind, because I just got a note in my chat from Miss Emily Peel, who's from the student and postdoc training group, just to let folks know that if the students or postdocs are meeting during the lunch break, and that'll be a virtual meeting, and she will send the Zoom link to myself or Tom, and then we'll place that in the chat for folks to chat. Okay. Thank you again, Tom.
Thank you very much. So today's symposium is entitled Stress Responses in the Vertebrate Forebrain, organized by me, but I have to thank a number of people. First of all the speakers, because when I put together the idea for this symposium, I just put together the idea for the symposium and a list of potential speakers without ever checking with them. And every single one of them said yes when I then checked, would you like to give this presentation? So that was very easy and very nice. We had one speaker who did have to, for personal reasons, have to drop out at a late date, and you've seen the change in the program, and I'll point that out in a little bit as well. I'd also like to thank Grace Capshaw for this nice logo, which I could never have done. Beautiful. And the program committee who picked this particular symposium proposal. So that's Andrew Ivanyuk, Chris Heasey, and Darcy Kelly. So why stress responses in the vertebrate forebrain? Over the last 10 years or so, I've more and more gotten inspired by, entangled in, however you want to say it, the animal welfare science community. And there is a big demand in animal welfare science for more neuroscience, essentially. So people who are interested in how you make an animal's welfare as positive as possible, and that could be farm animals, that could be lab animals, that could be pets, that could be zoo animals, any of those, are interested in understanding when an animal is in a good state, when an animal is in a bad state. And that is not an easy thing to measure. And one potential approach to that is to know more about the nervous system and measure aspects of the nervous system which might reflect the animal's actual experiences. And so that brings up a whole number of questions, of course, is what would you look at? How do animals respond to good and bad environments? And in fact, are those responses similar across species, or are they very different across species, et cetera, et cetera? So there is a big interest in subjective experiences, obviously a very, very difficult topic to approach in any non-verbal creature. But there is at least some argument to be made that if there is such a thing as a subjective experience, the brain must be processing some of that. And if we can understand a bit more how the brain does these things, we might be able to measure brain-related parameters that might give us some insight into the animal's experiences. Because more and more species are also coming under the umbrella that people are interested in. It's not just dogs and cats and horses. It is frogs, it's fish, it's these days even invertebrates, many types. It's becoming more and more complicated. And I went to a meeting last year in London, and that the very purpose of that meeting was to try and understand whether there were measures of welfare that could be developed that could be practical and applicable across many species rather than just for individual species. And it's a big ask, but the way, of course, to approach is to have a comparative evolutionary phylogenetic approach, because that's the only way we're going to well understand and map similarities and differences between species. So that's where the inspiration for this symposium came from. This focus on stress is because we still don't really know much. There's still a lot to know. I shouldn't say it the other way. We know a lot about stress, but there's also a lot we don't know yet. And of course, chronic stress is one of the major factors that would relate to negative welfare in animals and in fact in people, of course. Chronic stress is a major risk factor for many mental health disorders, etc., in humans as well. And the response to this chronic stress is not just in the hypothalamus, it's not just in the brainstem, it is also in the forebrain. There's many aspects of the forebrain that are involved in the assessment of the situation, in therefore deciding whether to activate certain stress responses or not, etc. So the forebrain processes presumably subjective experiences. Now we're talking vertebrates alone, of course. Other structures may be doing this in insects. That's a bit beyond the topic of today's symposium. And so the forebrain responds to stress and regulates stress responses. And so therefore, looking at how the forebrain in different vertebrates regulates and responds to these stresses is of interest to this bigger picture question. Already said, a comparative approach is very useful. First of all, because we can actually use information from humans and relate that to other species, because in humans we have the unique data, at least, of getting verbal responses about subjective experiences, which we cannot get from any other creatures. So if we can relate those verbal responses to human brain activity and then relate that human brain activity to other species, that might give us insights into these other species in a way that we couldn't without the humans, but also the other way around, of course. The more we know about how common and similar something is across species, the more likely it will also be similar in humans and vice versa. So we can find where are the things that we really cannot go beyond the species because it's very species specific, and what are the things that are quite general so that we can actually translate it from one species to another. And so that may then allow us to find, in an animal welfare context, good markers, neural markers of the experience of negative experiences like chronic stress, but also how we might make the animals and people, of course, more resilient to stress. So what distinguishes individuals that are more resilient from those that are not? And we have a talk about that later this afternoon. And so, of course, we're all interested in evolution as well. So the understanding of the evolution of the forebrain and how it relates to, for example, the hypothalamic stress regulation is in itself, of course, also very interesting to see if this is a very anciently conserved property or if this is indeed something that has evolved a few times in different vertebrate lineages. So today, after my introduction, we're only running five minutes over right now, but that's because we started 5 minutes late, so we're perfectly fine on time. We will have 3 presentations by Marco Vindas, David Valaye, and Loretta Medina before the break. Every slot is 35 minutes. I've asked the speakers for a 25-minute presentation and 10 minutes of for questions at the end of that. We have talks about fish, about pigs, about birds and mammals more generally, and about more birds at the end as well. So we take a 15-minute break after the first 3 talks so that people can stretch their legs, get a little, get their eyes off the screen. And then we have 2 more talks before the food break. I call it a food break because here in the UK, that's dinner. But if you're on the East Coast of the US, I'm assuming that will be lunch for you. And if you're on the West Coast of the US, I'm afraid that's going to be the middle of your morning, and lunch will be when we start up again, which is about 1 hour and 45 minutes later. So it is a very long break. That's partially because one of the speakers dropped out, and so that would have made for a slightly shorter break if she had been able to speak. But I didn't think anybody would mind to have 1 hour and 45 minutes to have their food. And then we'll have 2 final speakers after the food break. Seema, who's in the US, so this is a better time of day for her anyway to speak. And then Carrie, who is currently in the UK, so she'll be speaking quite late in her own time zone. And then we'll have some time at the end for general discussion if people wish that, and we'll finish up by about, 9 o'clock over here, 3 o'clock in the afternoon in Chicago, I guess, if to pick a random time zone. So without further ado, I'll stop sharing and like to invite Marco to share his screen and present us with his presentation, please.